Hello students, welcome to the biology class. Today we are going to continue with our chapter Respiration in Plants. In our previous sessions we discussed about the glycolysis, Krebs cycle, and today we are going to discuss about electron transport system. So what is electron transport system or electron transport chain is the final stage of aerobic respiration by which electrons derived from various substrates are transferred to oxygen. It is a series of highly organized oxidation reduction enzymes. So, respiratory breakdown of glucose in the presence of oxygen is an oxidative process. During this process, several intermediates are oxidized. The energy-rich molecules like NADH2 and FADH2 released during the glycolysis and Krebs cycle undergo oxidation, releasing energy, and finally the water is formed. So this is called as terminal oxidation. The oxidation of NADH2 or FADH2 occurs by transfer of their electron to a series of electron acceptors, and finally they combine with water to form they combine with oxygen to form water and ATP, that is the energy. The metabolic pathway through which the electron pass from one carrier to another is called electron transport system or electron transport chain. Electron transport system, electron transport chain is present in the inner mitochondrial membrane. Components of electron transport system or electron transport chain. Various components of ETS are arranged in following complexes. So there are 5 complexes. Number 1 is NADH dehydrogenase complex. Complex 2 is succinate dehydrogenase complex. Complex 3 is cytochrome BC1 complex. Complex 4th is cytochrome oxidase complex, and the 5th is ATP synthetase complex. In this image you can see that is the electron transport chain present in the inner mitochondrial membrane. So these FMN, FES, this UQ means ubiquinone, UQH2 means that is reduced ubiquinone. These are your cytochrome B, that is again FES, cytochrome C1, passes from this, they passes again to the FAD, then FES, then cytochrome A, cytochrome A3 and copper ions. So these are your electron acceptors and this is known as your electron transport chain. This cytochrome C and cytochrome C, they are known as the mobile carriers. This is the matrix where the Krebs cycle takes place, and the first complex, that is NADH dehydrogenase, the second complex, that is succinate dehydrogenase, the third is cytochrome BC1, fourth is your cytochrome C oxidase, and the last and the fifth is your ATP synthetase, that is on the oxysome, that is known as FOF1 complex. So this is the inner membrane space, but the electron transport membrane, that is the electron transport system, takes place in the inner mitochondrial membrane. Mechanism of electron transport system. The first is electron transport system is a metabolic pathway through which the electron pass from one carrier to another. NADH2 are produced in the mitochondrial matrix during glycolysis and citric acid cycle are oxidized by NADH2 dehydrogenase. Oxidized means that hydrogen ions, they are being released, and electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives the reducing equivalents via FADH2 that is generated during oxidation of succinate through the activity of enzyme succinate dehydrogenase, and that is complex 2 in the citric cycle. The reduced ubiquinone, that is ubiquinol now, is then oxidized with the transfer of electrons to the cytochrome C via the cytochrome Bc complex, that is complex 3. Complex 4 refer to the cytochrome A and A3 and 2 copper ions. Then when the electrons pass from one carrier to another via complex 1 and 4 in the electron transport chain, they are coupled to ATP synthetase, that is your complex 5th, for production of ATP from ADP and inorganic phosphate. During the electron transfer, the hydrogen atom splits into protons and electrons. The electrons are carried by the cytochromes. They recombine with protons before the final stage when hydrogen atom is accepted by molecular oxygen to form the water known as terminal oxidation. So once again go through this diagram, that is your first complex, and NADH is oxidized to NAD. Hydrogen ions, that is going to be splits into the electrons and to the protons. So electrons, they are being accepted by the electron acceptor, that is your electron transport chain, to the ubiquinone, and the protons, they enter in the intermembrane space. Similarly, in the complex 2, when FADH2 is oxidized to FAD and FES, then the hydrogen ions, they are being passes as a protons into the intermembrane space. Electrons are accepted by the electron acceptors, and then there is a complex 3. Again, the electrons are accepted by the electron transport chain, that is cytochrome C, and the hydrogen ions, which is splits into the protons, they passes into the intermembrane space. And similarly with your complex 5th, and here the hydrogen ions, that is being accepted by the oxygen, that is the terminal position, and they form the water. So this is known as the terminal oxidation. Now, as the proton ions, that is going to be increased in the intermembrane space as compared to the matrix. So the concentration of protons in the intermembrane space is increased, so that causes the gradient, means the difference in the concentration. So to maintain the equilibrium, these protons, they have to shift it back to the matrix. But for coming back from intermembrane space to the matrix, the route is only through the complex 5th, that is coupled with ATP, that is your oxysome made up of 2 units, that is F0 and F1, and F1, that is having the ATP synthetase. So when that particular protons, they are coming back, so the energy of that will cause the release of ATP synthetase enzyme, and because of that enzyme, the ATP is being synthesized, which is the end product of aerobic respiration. Water is formed in the complex 4th, ATP is formed in complex 5th. So this is the end of your aerobic respiration, and the process is known as oxidative phosphorylation. So now we will discuss what is oxidative phosphorylation. The synthesis of ATP during the aerobic respiration is termed as oxidative phosphorylation. The oxygen is here used as oxidant and reduced coenzymes are oxidized by electron transport process and coupled to ATP formation. Mechanism of oxidative phosphorylation. During oxidative phosphorylation, the protons are returned to the matrix from higher concentration of proton outside the inner membrane. The energy released by flow of protons during the gradient is utilized in synthesizing the ATP with the help of ATP synthetase, the complex fifth. This complex consists of two major components, that is F1 and F0. The F1 headpiece is peripheral membrane protein complex and contain the site for the synthesis of ATP from ADP and inorganic phosphate. So this is the diagram, very important diagram. So this is your oxysome, that is F0 and F1. So this is the peripheral region present in the matrix and this is present in the inner mitochondrial space. So the protons, while they are coming back from the outer space to the matrix, the energy is released, and because of that energy, that is the synthesis of ATP synthetase, and that leads to the formation of ATP, which is known as oxidative phosphorylation. So FO is an integral membrane protein complex that forms the channel through which the protons cross the inner membrane. When two hydrogen ions, means when the protons pass through this, the energy contained in them is used for the synthesis of one ATP molecule. For every two hydrogen ions that pass through F0 from the intermembrane space, two ATP molecules are produced. So each NADH2 molecule is oxidized to 3 ATPs and each FADH2 into 2 ATP molecules. Oxygen acts as a terminal acceptor of hydrogen to form the water and drives the whole process by removing the hydrogen from the system. So this is how the oxygen is released, water is formed, and the energy is synthesized during the aerobic respiration. Our next lesson and the last lesson of this chapter is an amphibolic pathway of the respiration, which we will discuss in our next session. Till then, stay home, stay happy. Thank you and have a nice day.